Hey guys, welcome back to One Stop Biology. So today we are going to discuss the very last video of Principles of Inheritance and Variation, which is the first unit of genetics. Basically, it is from class 12th NCERT Biology. So we are going to discuss all about the genetic disorders. Guys, before I start off with the topic, I would just request you to definitely like the video if you understood it well after the video gets over, and please do let me know in the comment section all about your feedback back about the video, whether you like it or you have any doubt or you want to improvise on anything. Please remember that your feedback is very, very important to me, and I would be waiting for your comments. So let's start off with the genetic disorder. The very first topic of genetic disorder that we are going to discuss is the pedigree analysis, right? So basically, what happened is that since a long time there was an idea about some disorders which are inherited, right? So after the rediscovery of Mendel's work, after the rediscovery of the work that Mendel did for us, what happened is people started analyzing the inheritance patterns in humans, right? And it was evident, very clearly evident, that there are certain genes which are inherited, and you know any abnormality in those genes resulted in abnormality in the later generation, right? So basically analysis of traits in a several generations of a family, what people did is they analyzed these traits in several of generations of a family, and the study of this is basically known as pedigree analysis. So basically analysis of traits in a several of generations of a family is nothing but pedigree analysis. So in pedigree analysis, basically in human genetics, pedigree study provides a very strong tool. Now, basically, it is utilized to treat inheritance of specific trait abnormality or a disease, right? So basically, what happened is that in pedigree analysis, the inheritance of a particular trait is represented in the family, and that is also in the family tree over generations, right? Now, if I show you some of the symbols which are used in pedigree analysis, these are some of the symbols which are used in human pedigree analysis, right? So if it's a square box, it is male. This is considered as male. If it's a circle, it is a female. And if it is something like this, it is sex unspecified. If it is red in color, it is affected individuals. This is for mating. This is for mating between relatives, right? This is parent. So these are the parents and these are the kids. These are the children, right? And this is in the order of birth, left to right. So this is the elder one, this is the younger one, right? Now this is, again, parents with male child affected with disease, right? If I write this, these are basically 5 unaffected offspring. So there are certain symbols which are used in the pedigree analysis, right? Now, basically, what we saw that in this chapter that genes are located on the DNA which are present in the cream chromosome, and this DNA is the carrier of genetic information. That is why it is said that it is transmitted from one generation to the other without any change or alteration. However, if there is any change or alteration in the DNA, right, that is reflected in the later generations, that might be reflected in the later generation as well. And these genetic changes are known as mutation. The alteration of genetic material is known as mutation. And there are a number of disorders which are found in human beings which are associated with inheritance of these changed or altered genes. And to study that, we use pedigree analysis, right? Now, the second topic is of Mendelian disorders. So, basically, the genetic disorders can be grouped into two categories. One is Mendelian disorder and the other is chromosomal disorder. Mendelian disorders are mainly determined by alteration or mutation in the single gene. Remember, Mendelian disorders are mainly determined by changes in the single gene. Now, these disorders are transmitted to offspring on the same lines. Remember that these are transmitted to offspring on the same lines and these are traced by pedigree analysis. For example, hemophilia, cystic fibrosis, sickle-cell anemia, color blindness, phenylketonuria, or thalassemia. All these disorders are nothing but Mendelian disorders, which is basically disorder, or basically this is determined by alteration of a single gene. Now, Mendelian disorders may be dominant or recessive. It might reflect or it might not reflect, right? So if you see the pedigree analysis here, you will see the first one is autosomal dominant trait, right? So this is the case in myotonic dystrophy, right? Wherein you see that the female has the disease here, right? In the case of sickle cell anemia, however, the parents seem to be normal, they are not affected. So that means that particular gene which is going to give the disorder is recessive in the case of both the parents. But however, you see that in the first generation, you will find progeny affected by that particular alteration in the gene, right? Now, the first type of Mendelian disorder that we are going to discuss is color blindness. Color blindness, guys, is a sex-linked recessive disorder. It is basically a defect which is either red or green cone of the eye, right? So it is a defect in either red or green cone of eye, which results into failure in discrimination between colors, right? So this defects basically makes someone to not to discriminate, basically failure to discriminate between red and green color. Now this defect is due to mutation in a certain gene which is present in the X chromosome. Remember that this defect is due to mutation in a certain gene present in X chromosome. Now it happens more often in females than in more often in males than in females. In the case of male, it occurs in about 8%, and in the case of female, it occurs in just about 0.4%. It occurs in about 8% of males and 0.4% of females. Now, this is because the gene that lead to red-green color blindness are on the X chromosome, right? Now, males have only one X chromosome and females have two, right? So, the son of women who carries the gene, remember what I'm saying, hear it very clearly. The son of a woman who is carrying a gene has 50% chance of being colorblind, right? The mother is not herself colorblind because the gene is recessive. So the mother is a carrier but is not herself colorblind. That means the effect is suppressed by her matching dominant normal genes because the mother has two X chromosomes. So the other normal gene will suppress the recessive one, right? So the daughter will normally be not be colorblind until and unless her mother is a carrier and father as well is colorblind, right? So this is all about color blindness. Now the second type of Mendelian disorder is hemophilia. Hemophilia again is a sex-linked recessive disease. Remember that all these are recessive diseases. Now, it shows its transmission from unaffected carrier female to some of the male progeny. Again, carrier is female. Now, in this disease, what happens is there is a single protein that is part of cascade of proteins which is involved in clotting of blood. And this protein is affected. And that results in non-stop bleeding from a simple cut, right? Even if there is a very simple minor cut, your blood is not going to clot and you are going to suffer from non-stop bleeding, right? So what happens here that the heterozygous female, which is a carrier, may transmit the disease, right? Now they transferred the disease to sons. Now the possibility of a female becoming a hemophilic is extremely rare because for that, mother of such a female has to be at least a carrier and the father should be hemophilic, right? Which is very difficult. Now the so basically hemophilia is attached to a very very royal name, Queen Victoria. So the family pedigree of Queen Victoria shows a number of hemophilic descendants because Queen Victoria herself was the carrier of the disease, right? So again, it is a recessive disease, sex-linked recessive disease caused by female. Females are the carrier, and it is passed to sons, not daughters, until and unless daughter, as in the mother of that daughter is a carrier and father as well is a hemophilic, right? So the chances of that happening is very less. Now the third type of Mendelian disorder is sickle cell anemia. This is an autosome linked recessive trait. Again, it can be transmitted from parents to offspring when both partners are carrier for the gene. So this happens when both the partners are carrier of the for the gene or heterozygous. Now, disease is controlled by a single pair of allele, which is HbA and HbS, right? So, out of the three possible genotypes, only homozygous individuals show the diseased phenotype. Heterozygous individuals, they are unaffected, but they become the carrier of the disease, right? So, if it is homozygous, so if you see here, if all three genotypes are HbS, then only you have sickle cell anemia. If you are heterozygous and you have just one HbS, then you become carrier of the disease, right? This disease is caused by substitution of glutamic acid by valine, right? And this happens at sixth position of beta globin chain of hemoglobin molecule. It is basically a hemoglobin molecule related disorder. Now, substitution of this amino acid in globin protein results to single base substitution of sixth codon of beta globin gene. Now, from GAG it changes to GUG, right? So if you see it here, these are normal hemoglobin cells, right? You have CTC, normal hemoglobin, and GAG. This is the sickle cell gene. Here what is happening is you have CAC and GTG. Now this GTG changes to GUG. And here if you see here, this is changing to the glutamic acid is being changed to valine because of this change in mRNA, right? And the entire sequence now is changed because you have kind of changed because now the product that it is going to produce will not be a normal hemoglobin gene, right? So this is sickle cell anemia. Now, what is happening here that the mutant hemoglobin molecule undergoes polymerization under low oxygen tension and that changes the shape of RBC. If you see here, RBC is biconcave disc, right? It changes the shape of RBC from biconcave to elongated sickle-like structure, and that is why it is known as sickle cell anemia. Now, the fourth type of Mendelian disorder is phenylketonuria. It is again an inborn error, and this is an inborn error of metabolism, and it is also inherited as autosomal recessive trait. Now, here what happens is that the affected individual lack an enzyme that converts the amino acid, which is phenylalanine, into tyrosine. Right? So what is happening is that phenylalanine is not being converted into tyrosine. And as a result of this, phenylalanine keeps on accumulating. Right? And that then if it starts accumulating, it gets converted to phenyl pyruvic acid. It starts converted into what? Phenyl pyruvic acid and other derivatives. Now, accumulation of these in brain, these are getting accumulated in brain. So accumulation of these in brain results in mental retardation. These are basically excreted through urine because of poor absorption by kidney. Kidney is not able to absorb it, right? So there are again multiple problems which starts happening because of accumulation of these phenyl pyruvic acid, right? So this is phenylketonuria, again a Mendelian disorder. The last Mendelian disorder that we are going to discuss is thalassemia. Thalassemia again is an autosome linked recessive blood disease, right? It is transmitted from parent to offspring when both parents are unaffected carrier for the gene. So it happens when both parents are unaffected carrier for the gene. That means that they both are heterozygous, right? The defect could be due to either mutation, so it can be due to mutation or deletion, which results in reduced rate of synthesis of one of the globin chains. So this results in reduced rate of synthesis of what? One of the globin chains. Alpha and we have alpha and beta chains, right? Which make up the hemoglobin. So now the synthesis rate of this globin chain is reduced. Now this causes formation of abnormal hemoglobin molecules, which results in anemia, right? So this can be basically classified according to which chain of hemoglobin molecule is affected. If the alpha chain is affected, we call it alpha thalassemia. So here alpha globin chain is affected. In beta thalassemia, the beta globin chain is affected, right? Now, in case of alpha thalassemia, it is controlled by two closely linked genes, which are HbA1 and HbA2 on chromosome 16 of each parent. Remember that alpha thalassemia is controlled by two genes, HbA1 and HbA2, which are present on chromosome number 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. In the case of beta thalassemia, it is controlled by a single gene, which is HBB. This gene is present on chromosome 11 of each parent. This is present on chromosome 11 of each parent, and this occurs due to mutation of one or both the genes again. So this is thalassemia. Now, the second type of disorders are chromosomal disorders. So basically, chromosomal disorders are caused by absence or excess or say abnormal arrangement of one or more chromosomes. Mendelian disorders were because of changes in one type of gene, right? So failure of segregation of chromatids during cell division, right? That results in gain or loss of a chromosome and is known as aneuploidy. It is known as aneuploidy. Basically, here we are talking about failure of segregation of chromatids during cell division, right? In the same manner, failure of cytokinesis after telophase stage of cell division, that it results in increase in number of a whole set of chromosomes in an organism. This is known as polyploidy. So there can be aneuploidy where there is a gain or loss of one chromosome. there can be polyploidy, whereas where there is an increase in a whole set of chromosomes. Now we know that the total number of chromosomes in a normal human cell are 46, 23 pairs, right? Out of which 22 pairs are autosomes and one pair is a sex chromosome, right? Sometimes what happens is their individual may be included or lack one pair of chromosome. So the first one is Down syndrome. Now, Down syndrome is a genetic disorder where there is a presence of additional copy. There is a presence of addition, an additional copy of chromosome, which is known as trisomy. Now, the additional copy of chromosome number 21. So this is a genetic disorder wherein chromosome number 21 is present in addition, right? So this was first described by Langdon Down. It was described by Langdon Down. And what happens here is that the affected individual is one short statured, right? So they have a small round head, furrowed tongue, and partially open mouth. They are short statured. The palm of the baby is broad into characteristic palm creases. So there are characteristic palm creases present. And physical and mental development is retarded. So physical, psychomotor, and mental development is retarded. So if you see the diagram here, basically what is happening is you have a flat head, you have a broad flat face, you have many loops, palm crease, you have big wrinkled tongue, you have congenital heart disease, and then you have physical, psychomotor, and mental retardness, right? Now, the second type of disease is Klinefelter syndrome. So, what is Klinefelter syndrome? In this, what happens is that you have an additional copy of X chromosome present. Now, these are the sex chromosomes, remember. So, you have instead of 46, 47 chromosomes, wherein instead of XY, you have XXY, right? So in case of male, what happens is that there is an individual who has an overall masculine development because they have XY, but then they have an extra feminine development as well, like development of breast, right? Now, these individuals are sterile. Remember that they are sterile. They cannot produce an offspring, right? They have extra development of feminine. They have extra feminine development, like development of breast. In the same way, you have Turner syndrome. In Turner syndrome, what happens? Yeah. So in Turner syndrome, what is happening is that there's an absence of X chromosome. Now, this happens in the case of females. So instead of 46 chromosomes, you have 45. So basically, usually a female has a pair of X, right? In this case, they have XO. So one X chromosome is absent. Now, again, such females are sterile. They are not able to produce offspring. Now, this happens because in this case, what happens? Ovaries are rudimentary. And then there is lack of secondary sexual characters. So if you see this diagram here, what you'll realize here is that in the case of Klinefelter syndrome, the male has features like developed breast, right? However, in the case of Turner syndrome in females, there is a short stature and the feminine characters, the secondary sexual characters are underdeveloped, right? So they'll have partially developed breasts. The ovaries are rudimentary, and in both the cases, these individuals are sterile. They will not be able to produce an offspring. So, guys, with this, we have finished the chapter of principles of inheritance and variation. We have covered all the genetic disorders that are given as per the syllabus of NCERT, as per the depth that is given in NCERT. I hope you would have understood the entire chapter and this video very well. Please do not forget to like the video and share it with your friends. If you are new, please do not forget to subscribe to the channel. And if you have any doubt, either mention it in the comment of this video, or you can also message me over WhatsApp, and the WhatsApp number is given in the description of the video. I'll be waiting for your comments, guys. Bye bye.